Hi, I'm Roger Deal with the Fred Hutchinson Cancer Research Center in Seattle, Washington. I'd like to tell you about a paper by Stephen Henikoff and I that was published in Developmental Cell. In this paper, we describe a new method for isolating nuclei from specific cell types of a whole tissue. This allows us to examine global gene expression and chromatin profiles in specific cell types of a whole organism. A major limitation to understanding the differentiation and function of various cell types during development has been a lack of straightforward methods for isolating specific cell types from whole tissue. Current methods for achieving this end, such as laser capture microdissection, fluorescence-activated cell sorting, have a variety of drawbacks that limit their general applicability. For example, both of these methods require harsh and extensive tissue manipulation. They generally provide low yields at less than optimal purity of the desired cell types. Furthermore, they both use instrumentation that's highly sophisticated and expensive and requires specialized technical skills to operate. Given this situation, we wanted to develop a new method that would be easier to use and would be approachable by all researchers in the field. Here's what we came up with. The basic premise behind our method is that in order to perform gene expression and epigenome profiling, we don't actually need the whole cells, but rather just the nucleus. So our strategy is to affinity label the nuclear envelope surface in the cell type of interest, but not in other cell types. In this way, we can prepare an extract of total nuclei from the tissue and simply select the nuclei that we want with an affinity selection. These nuclei can now be used not only for chromatin profiling, but also for gene expression profiling. We and others have shown that the nuclear RNA pool is basically comparable to the total RNA pool, and so we can get accurate gene expression profiles from nuclear RNA. We call this method INTACT, for isolation of nuclei tagged in specific cell types, and there are two components to this system. One is a cell type specific promoter or enhancer trap that's expressed in the cell type of interest and not in nearby cells. And the second part is an affinity tagged nuclear envelope protein. And for this we use a three-part fusion protein consisting of a nuclear envelope targeting domain from the RAN GTPase activating protein, GFP for visualization, and the biotin ligase recognition peptide, which in the presence of E. coli biotin ligase becomes biotinylated. And therefore, nuclei that have this protein on their surface can be purified by virtue of the interaction between biotin and streptavidin. Here's how the procedure works. We begin the procedure by making a crude nuclei preparation from our transgenic organism in which a cell type of interest has affinity tagged nuclei. We make this extract in a small volume to which we add magnetic streptavidin-coated beads to allow them to bind to the labeled nuclei. We then mix this solution for half an hour in the cold room to allow the beads to bind to the nuclei. After this, we increase the volume to get better separation between the bound nuclei and other particles in the mixture, which allows for a more efficient purification. Once we've diluted the bead nuclei mixture, we draw it up into a 10 ml serological pipette and allow it to flow past a magnet to collect the bead-bound nuclei. And we do this by inserting the serological pipette into this homemade magnetic purification device, which consists of a magnet with a 1 ml pipette running through the slot in it. We allow this solution to drain at a slow flow rate, and as it drains, the bead-bound nuclei and free beads will stick to the wall of the pipette tip. Once the column is completely drained, we can then dismantle it in order to elute the bead-bound nuclei from the wall of the pipette tip. We do this by simply inserting the pipette tip onto a 1 ml pipette and using it to draw in and out a fresh solution of nuclei purification buffer. In this way, we get the beads off the wall of the tip and into solution again. And now we have a pure population of nuclei from our cell type of interest. As a proof of concept, we set up the INTACT system to to measure global gene expression and chromatin profiles in each of the two cell types of the Arabidopsis root epidermis, hair cells and non-hair cells. Although each of these cell types represents only about 10% of the total cells in the root, we could routinely get several 100,000 nuclei from each, starting with just a few grams of tissue. And in each case, we get essentially 100% pure nuclei. Using this method, we identified several hundred genes that are expressed preferentially in each of these two cell types, and we could identify epigenomic differences that are associated with cell type specific expression. These data will now serve as a platform for further investigation of the function of these two cell types through reverse genetics and other similar approaches. In conclusion, our paper shows that the intact system outperforms existing methods in terms of ease of use and the purity of the cell types of interest. Importantly, this method should be adaptable to any organism that can be genetically transformed. We hope that you'll find the intact system useful for your own research, and at the very least, enjoy reading the paper. Thank you.